Fenomenal.
Good morning. First of all, I would like to thank all of you for attending this webinar. The webinar today is from the group of molecular bases of pathology, and the coordinator is Patricia Aspichueta. Patricia, thank you for organizing this meeting. And if you want, you can introduce the speaker today.
Yes, of course. Thank you, Mar. Well, it's a great pleasure for me to be here and to introduce Pedro Rodrigues. He is a PhD in pharmacy from the University of Lisbon in Portugal in 2018. He joined the Liver Disease Lab led by Dr. Bañales. He's now an IKERBASQUE research fellow and a Sarah Borrell researcher. He has published several journals in high impact journals in the area of pathology and gastroenterology. He has also received several awards. In fact, this year he has just received the UEG Rising Star from the European Gastroenterology Union Science Association, which distinguishes young emerging leaders in the field of gastroenterology. He is a PI or has been a PI in several national and international projects. He is also a management committee member in the European network from the study of cholangiocarcinoma and in the Euro-CholangioNet Cost Action. He has supervised several research works, master students, PhD students, and well, he is young and he is an emerging leader in the field of pathology and gastroenterology. So thank you, Pedro, for being with us. And today he's going to tell us about MIR21 and non-alcoholic fatty liver disease.
So good morning all. Thank you very much, Patricia, for this kind introduction, and thank you very much to the Spanish Association for Biochemistry and Molecular Biology for this kind invitation. It's really a pleasure for me to be here. So I'll share my screen. Okay, I think everyone is now watching it. So basically today I will talk a little bit about my PhD work that I was developing in Lisbon, that was later on continuing during my postdoc studies now. And I will talk a little bit about the role of microRNA 21 in non-alcoholic fatty liver disease pathogenesis. Sorry. So nowadays, the unlimited access to caloric dense food in combination with the lack of physical activity has been largely contributing to the increased prevalence and incidence of metabolic liver diseases, particularly non-alcoholic fatty liver disease. And for instance, as we can see here in this map, the prevalence of non-alcoholic fatty liver disease reaches an average of 25% of the population, which is different from the countries, but is currently an alarming problem worldwide. What is then non-alcoholic fatty liver disease? Non-alcoholic fatty liver disease comprises a spectrum of liver lesions ranging from the simple accumulation of liver in the fat, also known as steatosis, to the progression of non-alcoholic steatohepatitis that might progress to liver cirrhosis and eventually to the development of hepatocellular carcinoma. It is currently the most prevalent liver disease worldwide and strongly correlates with diabetes and the metabolic syndrome. The pathogenesis is currently not unveiled, and there are no efficient therapies to treat these patients. There are several risk factors that were already proposed to be linked with NAFLD. Among them, the two more risk factors that were mostly described is obesity, since 91% of patients with NAFLD are also morbid obese, and 37% of them have also NASH, and also insulin resistance, since 47 to 90% of patients with NASH are diabetic. Still, the exact mechanisms underlying NAFLD pathogenesis and progression are not fully understood. Usually, NAFLD pathogenesis may start by disordered lipid metabolism. And what usually happens in the liver in this case, the accumulation of fat in the liver might occur due to three distinct processes. It may occur by the increased fat uptake that usually comes from diet. It may occur by an increased de novo lipogenesis that usually occurs due to hyperglycemic and hyperinsulinemic states, and also occurs by the lipolysis from the adipose tissue that markedly contributes to the accumulation of fat in the liver. This results in the promotion of accumulation in fat that promotes insulin resistance both in the liver and also in the skeletal muscle, that aggravates the insulinic resistance state in the adipose tissue, contributing to a vicious cycle of insulin resistance and accumulation of fat in the liver. Several factors were already associated with the pathogenesis of NAFLD, and from the progressions from simple steatosis to non-alcoholic steatohepatitis and to fibrosis and cirrhosis. One of these factors are microRNAs. What are microRNAs? MicroRNAs are small non-coding RNAs that regulate gene expression in a post-transcriptional level. Briefly, after transcription in the nucleus, the primary microRNA is processed by the Drosha complex, leading to the formation of the precursor microRNA, which is then exported to the cytoplasm through the exportin 5 complex. Then, with the dicer complex in the argonaute, the precursor microRNA is processed, leading to the formation of the mature microRNA, which will lead to the repression of protein translation, either by inducing a deadenylation and degradation of the mRNA transcript, or even inducing endonucleolytic cleavage and direct degradation of this transcript. Therefore, microRNAs usually may decrease mRNA expression in a negative way. What do we know about microRNAs and metabolism? Several microRNAs were already reported to be involved in metabolism, not only in the liver, but also in other metabolic tissues. And for instance, microRNA 34A was shown to target sirtuin-1 and another metabolic relevant targets such as VAMP2 and BCL2, not only in the liver, but also in the pancreas, leading to insulin resistance. In this regard, our group has previously shown that microRNA 34A levels are progressively increased in the liver of patients with NAFLD from simple steatosis to less severe NASH, and progressively increased to patients with more severe NASH. This resulted in a progressive decrease in the protein levels of sirtuin-1 and the accumulation of acetylated levels of P53. And not only MIR34A are being studied in the field, but several microRNAs are now reported as circulating signatures in liver disease and also for NAFLD. Among them, MIR21 is being extremely studied in liver diseases and also in oncological disorders, since it was one of the first oncomirs described in cancer. And for instance, in patients with NAFLD and NASH, MIR21 was found to be increased in the serum of these patients, potentially harboring diagnostic value. But what about their role in liver disease? microRNA 21 is typically an oncomere that was previously reported to target several targets, regulating then invasion and metastasis, cell mortality, cell proliferation, invasion and metastasis, and also cell survival. Still, the role of this microRNA in NAFLD was not reported when we started our studies. And not only microRNAs are important molecules in NAFLD pathogenesis, but also bile acids. So what are bile acids? Bile acids are amphipathic molecules that are usually found in bile, produced by hepatocytes, that have an important role in digestion, in fat absorption. But they are also signaling molecules. And according to their chemical structure, they might be categorized or more toxic bile acids, such as deoxycholic acid or chenodeoxycholic acid, or in more cytoprotective bile acids if they are more hydrophilic, which is the case of ursodeoxycholic acid or UDCA. And for instance, the toxic bile acids may induce apoptosis through the extrinsic and intrinsic pathways of apoptosis. What do we know regarding bile acid levels in patients with steatohepatitis? Back to 2008, our group was involved in measuring the levels of bile acids in the liver of patients with non-alcoholic steatohepatitis. And we were surprised to observe that the bile acid pool in the liver of these patients, when compared with control, were mostly hydrophobic. And among the hydrophobic the bile acids, particularly the levels of deoxycholic acid increased almost 92% when compared with controls, which might represent a novel pathogenic effector and player in naphthalene pathogenesis. What are the mechanisms that are already known for DCA-induced apoptosis? As I mentioned, DCA is a toxic bile acid. It is well known to induce reactive oxygen species in hepatocytes, leading to the formation of genotoxic damage and the induction of apoptosis through the extrinsic and intrinsic pathways. And regarding the modulation of microRNAs by deoxycholic acid, we have previously shown that DCA induces liver cell death by activating the miR34A pathway in primary hepatocytes. And regarding miR21, we have previously shown that DCA were able to significantly reduce the levels of miR21 in primary hepatocytes when compared with controls, but still the mechanisms were not yet studied. Therefore, our first aim was to clarify the mechanisms by which DCA modulates this miR21 signaling pathway and how this pathway may contribute to apoptosis in primary red hepatocytes. So we firstly started to evaluate if DCA was able to modulate the miR21 expression, miR21 pathway in a dose-dependent manner. And we observed that the levels of miR21 significantly decreased with increased concentrations of deoxycholic acid in culture, while the levels of PDCD4, which is one of the targets of miR21 already validated, that are usually shown to be upregulated during apoptosis, markedly increased with the increased concentrations of DCA. And this resulted in an induction of cell death, as we can see here, and a reduction of cell viability. And this cell death was mostly apoptosis, since we observed that the levels of caspase 3 and 7 activity markedly increased with the increased concentration of DCA. And also, we can see here in these images, an increase in the number of apoptotic nuclei after incubation of DCA. So we decided to evaluate later what is the impact of this pathway in the induction of apoptosis by DCA. And to do that, we decided to overexpress microRNA 21 in primary hepatocytes. We observed that after overexpressing miR21, the levels of PDCD4 were reduced, and the ability of DCA to induce PDCD4 was markedly reduced, as we can see here in these bars. And importantly, DCA-induced cell death, as we can see here in these green bars, was reduced when compared with control conditions. Therefore, we may conclude that miR21 pathway, the reduction that we observe by DCA, is significantly contributing to DCA-induced cell death. So our next question was to what are now the mechanisms upstream of the modulation of miR21? And we found that that was in the time we were doing this study, it was recently reported that the promoter of microRNA 21 had an FKB response element. So we hypothesized maybe DCA might decrease miR21 expression by inhibiting NF-κB activation. So we measured NF-κB nuclear and cytoplasmic levels, and we were happy to observe that the nuclear levels of NF-κB were markedly decreased with increased concentrations of DCA, while the cytoplasmic levels accumulated, which indicates that NF-κB might be inhibited. But to confirm these results, we decided to measure the transcriptional activity of NF-κB by using a luciferase plasmid that contains the NF-κB responsive element. And we observed that when we incubated primary red hepatocytes with DCA, the NF-κB transcriptional activity was markedly reduced, thus confirming that DCA is actually inhibiting this pathway. But our next question was, but is this inhibition related with the inhibition of miR21? So to do that, we decided to overactivate NF-κB pathway using genetic approaches. We transfected primary red hepatocytes with either a constitutively active form of IKK, which is the enzyme that activates the NF-κB pathway, and also with NF-κB itself. And we were happy to observe that after activating NF-κB pathway, the DCA-dependent modulation of MIR21 was abrogated, and DCA-dependent apoptosis was decreased. And importantly, the levels of PDCD4 were also modulated, which clearly indicates that NF-κB is modulating MIR21 pathway when hepatocytes are treated with DCA. We also decided to evaluate if oxidative stress was involved in this process, since one of the mechanisms by which DCA induces cell death in hepatocytes is the induction of oxidative stress. So we confirmed that with the doses we were using, the levels of reactive oxygen species, or ROS, were significantly increased with increased levels of DCA. And to evaluate the relevance of reactive oxygen species in this process, we decided to use an antioxidant compound, N-acetylcysteine, or NAC, to evaluate the modulation of this pathway. And we incubated cells with N-acetylcysteines alone, DCA alone, or both in combination, and evaluated both MIR21 expression and DCA-induced cell death. And surprisingly, we observed that DCA-dependent modulation of MIR21 was reduced, and the DCA-induced cell death was reverted almost to basal levels, which clearly indicates a role of oxidative stress in this process. We next question, how can then oxidative stress might impact on this pathway? And how can oxidative stress modulate NF-κB signaling and then contribute to the modulation of the MIR21 pathway? And we came out with this pathway that was started to being studied in the beginning of the last decade, which is the PID-caspase-2 signaling. Briefly, what happens when facing cells when are facing a genotoxic stress, if these stress are considered a low-level stress, which induces a low-level DNA damage, PID, which is a protein that is induced by p53, is cleaved once, leading to the formation of PID-C. This PID-C usually may form complexes with NEMO and RIP1, which induces the activation of NF-κB, which allows cells to survive and try to repair the damage that was induced. On the other hand, when we are facing a high-level DNA damage, PID-C is then cleaved again to the formation of PID-CC, which forms a complex known as pidosome with RIDE and procaspase-2, which leads to the activation of caspase-2 and then the activation of apoptosis. We next decided to evaluate if there was a differential modulation of this signaling pathway in primary red hepatocytes treated with DCA. And we observed that the levels of PID-CC, the fragment that is cleaved twice and activates caspase-2, was markedly increased when cells were treated with DCA. And this results in the activation of caspase-2. So what is the relevance of this pathway, of this oxidative stress and PID-CC in the process? We observed that when incubated cells with an inhibitor of the oxidative stress, the N-acetylcysteine, the levels of PID-CC were reduced and the activation of caspase-2 were also reduced, which resulted in a decrease in the levels of EKBB levels, which indicates that the levels of NF-κB and the activation of this process was reduced. Then we confirm that this oxidative stress is actually engaging the two cleavages of PIDs, leading to the formation of PID-CC and to the inhibition of NF-κB, then modulating MIR21 pathway. We next decided to evaluate if this pathway also happens in vivo. And we treated rats by oral gavage with DCA during 1 and 3 days. And particularly after 1 day of gavage, we observed that MIR21 was markedly reduced, resulting in an increase in PDCD4, and also we observed a decrease in NF-κB activation and an increased levels in the cleavage of PID-CC and also in the activation of caspase-2. So we confirmed that this pathway also occurs when DCA accumulates in the liver of rats. So as conclusions of this first part, we can say that DCA modulates the MIR21 PDCD4 pro-apoptotic pathway in a dose-dependent manner. Briefly, we were able to show that DCA increases the levels of ROS, which results in the proteolysis of PID, leading to the consequent inhibition of the NF-κB pathway and the inhibition of the MIR21 levels, which results in the accumulation of PDCD4 and the promotion of apoptotic cell death. Moving on to the study of the MIR21 pathway, apart from studying the levels of and the role of PDCD4 in hepatocyte cell death, we came out with a newly recently described target of MIR21, which was PPAR-alpha. This receptor is mostly involved in the regulation of fatty acid oxidation. So, as I said, PPAR-alpha is a master regulator of lipid metabolism. It has a lot of functions in the liver. It modulates fatty acid uptake. It regulates mitochondrial beta oxidation, microsomal omega oxidation, and also the peroxisomal beta oxidation. It might also have an anti-inflammatory role by inhibiting the NF-κB pathway, and it also regulates SREBP1C. And importantly, it is already described that PPAR-alpha null mice that have no expression of PPAR-alpha in the liver, they have increased susceptibility to develop NASH. So we decided to evaluate, probably in NASH patients, MIR21 is regulating PPAR-alpha. And importantly, we also know that there are increased fatty acid levels in the liver of patients with NAFLD, and these increased levels of fatty acids might result in the promotion of MIR21, which will then inhibit PPAR-alpha and inhibit fatty acid oxidation, thus resulting in disease progression. So we decided then to evaluate this hypothesis, and our second game was to ascertain the contribution of the MIR21 PPAR-alpha signaling pathway in the pathogenesis of NAFLD. To do that, we generated in our lab MIR21 knockout mice, a whole organ knockout in all organs in mice, and we fed both wild-type mice and MIR21 knockout mice with a NASH-inducing diet, the MCD diet, for both 2 weeks to early NASH and for 8 weeks to more advanced NASH with fibrosis. And we were observed that macronutrient levels were significantly increased in MCD-treated mice, both at 2 weeks and 8 weeks. And this resulted in a decrease in PPAR-alpha levels, both in 2 and 8 weeks. And importantly, when evaluating the levels of PPAR-alpha in MIR21 knockout mice, we observed that these mice displayed increased PPAR-alpha levels when compared with the wild-type counterparts. When evaluating the liver histology of these mice, we observed that MCD induced accumulation of fat in the liver, a progressive one from 2 weeks to 8 weeks, and this accumulation of fat was clearly diminished in the liver of MIR21 knockout mice fed an MCD diet, which clearly indicates a potential role of this microRNA in the modulation of NAFLD pathogenesis. And we also decided to evaluate liver inflammation, and the levels of 2 important pro-inflammatory cytokines that play a role in NAFLD pathogenesis were markedly increased in wild-type mice and significantly decreased in MIR21 knockout mice, either at 2 or 8 weeks after MCD feeding. This indicates that the ablation of MIR21 knockout not only reduced the accumulation of fat in the liver, but also impacted on inflammation. We then measured liver injury, and we observed that the circulating levels of ALT, which is a serum marker of liver injury, were increased in wild-type mice with NASH, both at 2 and 8 weeks, and this liver injury was significantly reduced, particularly at 8 weeks, which shows that macronutrient knockout mice are clearly protected from liver injury. And this was accompanied by a decrease in the activation of caspase-2, which was clearly activated in wild-type mice and reduced in the MIR21 knockout mice treated with fed with the MCD diet. To confirm this data, we evaluated the levels of apoptotic cells in these mice, both at 2 and 8 weeks, and we observed that MCD induced an upregulation of the number of apoptotic cells in the liver, both at 2 and 8 weeks, and the number of apoptotic cells in MIR21 knockout mice was markedly reduced when compared with control ones, which confirms that these mice are protected from liver injury when developing NASH after MCD feeding. Regarding fibrosis, we observe that the expression levels of profibrogenic cytokines such as TGF-beta and the more common fibrogenic compound collagen 1-alpha-1 was significantly reduced in ME21 knockout mice when compared with the wild-type mice fed the MCD diet. And this resulted that all the wild-type mice developed a significant fibrosis after 8 weeks of feeding, while most of the ME21 knockout mice presented negligible fibrosis. There, we can conclude that the ablation of ME21 in mice with NAFLD and with NASH not only reduced steatosis, inflammation, and liver injury, but also resulted in a prevention of fibrosis development. We now decided to evaluate, because the ablation of ME21 results in the activation of PPAR-alpha, which is an important nuclear receptor in the pathogenesis of NAFLD, but there are also another promising nuclear receptors in the field of NAFLD, and one of them is FXR. So our next step was to evaluate if there might be a potential additive effect of the ME21 inhibition and PPAR-alpha activation, and also the activation of FXR using obeticholic acid. So what is FXR? FXR is a well-known bile acid sensor in the liver. The activation of FXR in hepatocytes usually induces the secretion of bile acids and also inhibits the synthesis. And also there are other roles of FXR in the liver, which is the modulation of lipid metabolism, the inhibition of inflammation, and also the potential inhibition of fibrosis. And importantly, it is already known that the activation of FXR clearly decreases the levels of triglycerides and cholesterol in the liver and in circulation. And most importantly, we know that FXR deficient mice develop steatosis and hypertriglyceridemia. Therefore, we decided to evaluate if the concomitant activation of FXR and PPAR-alpha by inhibiting ME21 might have a therapeutic role. And how can we activate FXR? We can use obeticholic acid. Obeticholic acid, or OCA, is a semi-synthetic derivative of chenodeoxycholic acid. This CDCA is the known endogenous activator of FXR. So this obeticholic acid is now in clinical trials for the treatment of NAFLD and was recently approved for the treatment of primary biliary cholangitis, and is a well-known FXR ligand that has strong anticholestatic, antifibrotic, anti-inflammatory, and hepatoprotective properties. So to evaluate the potential combination of ME21 ablation and FXR activation in mice, we fed wild-type mice and ME21 knockout mice with a standard diet supplemented with obeticholic acid, or with a fast food diet, or a fast food diet supplemented with obeticholic acid. This fast food diet was a diet, a novel NASH model that was reported years ago, that is mainly based on the Western diet characteristics that we are usually facing. It's basically a model based on the feeding of mice with a diet that is highly enriched in cholesterol, in fat. There is also drinking fructose in the water, and these mice are usually trying isolated to try to mimic the well-known sedentarism that we are now facing in the society. What we observed was that the wild-type mice fed with a fast food diet increased weight over the 6 months of diet, and this increase in weight was markedly reduced in the knockout mice fed with the fast food and supplemented with obeticholic, as we can see here in the green line. And this resulted in a marked reduction in the liver to body weight ratio, which was markedly reduced when compared with the wild-type mice. So basically, wild-type mice fed with a fast food diet developed hepatomegaly, and this hepatomegaly, this increase in body size, in liver size compared to the body weight, was reduced in the knockout mice fed with a fast food supplemented with obeticholic acid. What happened to MIR21 expression? We confirmed now in this model as well that mice with NASH display increased levels of MIR21 expression, which resulted in a marked reduction of PPAR alpha levels. And importantly, in the knockout mice fed with a fast food supplemented with obeticholic acid, the levels of PPAR alpha were markedly increased. What happened then in the histology of the livers of these mice? We observed that the wild-type mice fed with a fast food diet displayed a marked accumulation of fat in the liver, and both MIR21 inhibition or obeticholic alone reduced fat content, as we can see here in the lipidosis score, but this effect was more pronounced when we combined MIR21 genetic ablation and the activation of FXR with obeticholic acid. And this was also observed when we measured the cholesterol content in the liver of these mice that accumulated in wild-type mice and was reduced in the liver of the knockout mice fed with a fast food diet and supplemented with OCA. Regarding inflammation, we observed that the expression levels of several pro-inflammatory cytokines, such as TNF-alpha, IL-1 beta, IL-6, or TLR4, were increased in the wild-type mice fed with a fast food diet and reduced in the knockout mice fed with obeticholic acid. We then decided to evaluate the insulin signaling pathway in these mice, since, as you may remember, these patients, most of the NAFLD patients, also present insulin resistance. Since this is a fast food model that closely resembles the histological features of NAFLD patients, one mark of this model is also the development of insulin resistance. So what happened to insulin response under physiological conditions? When insulin binds to its receptor, insulin substrate receptor, it induces the phosphorylation of IRS in tyrosine residues. This results in the activation of PI3K and the phosphorylation of AKT. And the phosphorylation of AKT has two roles. It will firstly induce the translocation of vesicles that contain the GLUT transporter to the plasma membrane that will allow the entrance of glucose to the cells, and it also inhibits GSK3, which induces the production of glycogen in hepatocytes. But what happened in insulin-resistant states? In these patients, there is a high accumulation of fatty acids in hepatocytes, and not only fatty acids, but also TNF-alpha may induce oxidative stress. This oxidative stress induces the phosphorylation of JNK, which is a stress kinase that will phosphorylate IRS, but not in tyrosine residues. The IRS will now be phosphorylated in serine residues, and this results in an inhibition of the PI3K pathway, and in this case, AKT is no longer activated, which results in an inhibition of the translocation of the GLUT-containing vesicles to the plasma membrane, and an inhibition of glycogen synthesis, and even more, a production of glucose that will go out from the cell and go into circulation. So in this case, as far as insulin increases, there will be no response to insulin, which results in a clear accumulation of insulin in circulation, and also it will perpetuate the increased glucose levels in these patients. So what happened to this pathway in our mice? We clearly observe an activation of the GNK pathway in the wild-type mice fed with a fast food diet, and this phosphorylation of JNK was markedly reduced in the knockout mice fed with obeticholic acid. And going further in the pathway, we observed also this in the other mediators of the pathway. We measured the phosphorylation of IRS1, the substrate of insulin, and also the activation of AKT. And we observed that all of these proteins were less activated, as we can see here with the bars with the red line in the wild-type mice fed with a fast food diet, and this effect was totally prevented in the MIR21 knockout mice fed with a fast food diet with obeticholic acid. So we were able to observe that the activation of both nuclear receptors, PPAR-alpha, by inhibiting MIR21, and also FXR activation using obeticholic acid was able to restore hepatic insulin resistance. But not only the liver is the only organ affected in patients with NAFLD, but also there is a marked dysfunction in the skeletal muscle. So we then decided to evaluate if MIR21 was also increased in the muscle of these mice. And we observed that the levels of MIR21 were increased in wild-type mice fed with a fast food diet, which resulted in, again, in a reduction of PPAR-alpha. And what happened then in the insulin signaling pathway in the muscle? We were able to observe that these mice, the wild-type mice fed with a fast food diet, also developed insulin resistance in the muscle, and this insulin resistance was reverted when MIR21 knockout mice were fed with a fast food diet and obeticholic acid. So our strategy not only improves hepatic insulin resistance, but we were also able to revert muscle insulin resistance. So until now, we were only measuring the levels in mice. So what happened in human NAFLD? We measured the levels of MIR21 in the liver and in the muscle of patients with simple steatosis and patients with NASH. And we observed that MIR21 expression markedly increased both in the liver and in the muscle. And this resulted in the liver in a significant reduction in PPAR-alpha protein levels. Apart from their role in the liver, we also measured the levels of MIR21 in serum of these patients, and confirming previous results in another work, we were observed that MIR21 were increased in the serum of patients with NASH when compared with steatosis, which might then harbor a diagnostic value to evaluate the progression from steatosis to NASH in these patients. So as the conclusions from these second and third parts, we were able to show that the MIR21 PPAR-alpha pathway is increased in patients with NAFLD and also in mice with NAFLD. We observed that MIR21 knockout mice displayed less steatosis and inflammation and were protected against fibrosis development. And also, we observed that MIR21 ablation, genetic ablation, and FXR activation exerted a beneficial effect in mice, strongly ameliorating NAFLD development and progression. As you may remember, in the progression of NAFLD, we know that NASH has an increased risk for HCC development. And what do we know now about the development of HCC in patients with NAFLD? There is now a huge increase in the prevalence of NAFLD HCC. As we can see here in this graph, in the first years of 2000, the prevalence of HCC arising in a background of NAFLD were negligible, while, as we can see here in the orange bars, the prevalence of NAFLD-associated HCC is markedly increasing throughout the years, and it might actually represent a huge public health problem now. And this is also observed in this graph, since until now, the most causes of NAFLD, of HCC development in a chronic liver disease background were both viral hepatitis or alcoholic liver disease. But as you can see here, throughout the years, there is an alarming increase, as you can see here in this purple bar, in this purple curve, sorry, in the prevalence of HCC arising in patients with NAFLD, while the others are more or less stabilizing. So it seems now that in the upcoming years, there will be a huge problem with the development of HCC in patients with NAFLD. And in this map that was recently published in Nature Reviews two years ago, we can see here the prevalence of NAFLD HCC worldwide. And in some countries, we can see that the prevalence is reaching alarming levels, while the levels of HCC arising in other etiologies is markedly dropping. So this is currently an emerging health problem that we are now facing. And what do we know about the progression from NAFLD to HCC? Actually, we don't know that much. We know that there are several factors that might promote the progression of NASH to HCC. We know that the dysregulation of FXR is one of them. We know that the increase of ROS, oxidative stress, and insulin resistance that may occur as a result of the dysregulation of the microbiome might also promote NAFLD pathogenesis, and the chronic inflammatory state in these patients with an increased levels of IL-6, TNF, and STAT3 that are always observed in patients with NAFLD and obese patients might set the ground for the development of HCC, and not only the chronic inflammation, but also fibrosis and even genetics, because there are now some genetic polymorphisms that were related with the development of more severe NASH and increased risk of progression to HCC. All of these factors might cooperate in the progression from NASH to HCC. Still, the mechanisms are not totally understood. So our next question was, is microRNA also involved in the progression from NASH to HCC? So to do that, we firstly evaluated the Mi21 levels in a cohort of patients, including 200 obese patients from the San Sebastian cohort. All of these patients had a BMI greater than 30, and we correlated Mi21 expression with the histological hallmarks of NAFLD to observe if Mi21 might correlate with disease progression. And what we observed that levels of Mi21 were significantly increased with increased stages, histological stages of steatosis, with inflammation, and with ballooning. And this resulted with increased Mi21 levels in patients with a NAS score, which is a histological score used for the diagnosis of NAFLD, in patients with 5 to 8 score, which is a score for definite NASH in these patients, when compared with patients with a score below than 5 and in the threshold for the NASH diagnosis. We also analyzed the metabolomic profile of patients in these patients, and we stratified our cohort according to Mi21 expression levels in high and low levels. And we compared the liver metabolomic profile of patients with high and low expression. And what we observed, as we can see here in this heat map and in this volcano plot, there are several metabolites and lipids that were altered in patients with high expression of Mi21 when compared with low expression of Mi21. And most of them were amino acids, oxidated fatty acids, acylcarnitines, carbohydrates, carboxylic acid, and nucleosides and nucleotides. And outstandingly, most of these metabolites are also altered in patients with NAFLD and more altered in patients with NAFLD-associated HCC. Then, to evaluate the relevance of the Mi21 PPAR-alpha pathway in the progression of NASH to HCC, we now developed a new model, which is the CDAA model, which is a model that results in the progressive development of NASH, NASH with fibrosis, and preneoplastic nodules, and HCC. To do that, we fed wild-type mice and Mi21 knockout mice with a CDAA diet for 32 weeks or 66 weeks to induce either NASH or NASH and HCC. Regarding the 32 weeks, we observed that the wild-type mice significantly increased weight throughout time, as we can see here in the black bar and red bar, but the Mi21 knockout mice were markedly leaner. Again, in this model, we observed that the hepatic Mi21 levels were increased in mice with NASH when compared with controls, which resulted in a marked reduction of protein PPAR-alpha levels, which were increased in the Mi21 knockout mice. We next evaluated the levels of PPAR-alpha metabolic relevant targets, and we observed that the expression levels of some of them that are involved in beta oxidation and peroxisomal oxidation, and also in fatty acid transport, were reduced in the wild-type mice fed with the CDAA diet, which are the diet that induces NASH, while the levels of these targets were increased in the Mi21 knockout mice fed with the CDAA diet. To confirm the role of this Mi21 pathway in promoting beta oxidation, we next decided to evaluate the activity of the complexes of the mitochondrial respiratory chain to confirm that beta oxidation is actually being engaged when we are inhibiting MIR21. To do that, we measure the activity of the complex 1, 2, 3, and 4, and we observed that the activity of these complexes, or most of them, was reduced in mice with NASH fed with the CDA diet, while MIR21 knockout mice displayed an increased activity of these complexes when compared to the wild-type mice, which clearly indicates that beta oxidation is actually being engaged in MIR21 knockout mice, therefore explaining why we have less accumulation of fat in these mice. Now we moved forward in the pipeline of NASH development, and we fed the mice for 66 weeks and we evaluated tumor development. Firstly, we observed that the CDA diet markedly increased the accumulation of fat in the liver, while this accumulation was reduced in the MIR21 knockout mice. And not only the accumulation of fat, but also we again confirmed that the inflammation was markedly reduced in the knockout mice fed with the CDA diet compared to the wild-type mice. What happened with tumor development? We observed that the wild-type mice, all of the mice that were fed with the CDA diet, developed at least one perineoplastic nodule, with one of the mice developing a trabecular hepatocellular carcinoma, while, as you can see here in the perineoplastic numbers of the MIR21 knockout mice and here in this liver, most of the mice did not develop any tumor lesion. We confirmed that MIR21 was also increased in the non-tumoral tissue of mice fed with the CDA diet for 66 weeks, and the expression of MIR21 was further increased in the mice that developed hepatocellular carcinoma. We then evaluated the levels of some of the MIR21 targets, again, PPAR alpha, PDCD4, and CDK2AP1, which is a target now known to regulate cell cycle. And we observed that the levels of these targets were reduced in wild-type mice and restored in the knockout mice fed with the CDA diet. We then conducted a mouse liver cancer array to evaluate the regulation of several pathways related with the development of cancer, and we evaluated this array using wild-type samples from wild-type mice that were enriched in nodules, and also from MIR21 knockout mice from non-tumoral tissue and also with nodules. And we evaluated the expression of 84 genes involved in carcinogenic progression of NASH. We observed that several pathways were upregulated in the wild-type nodules. Some of them were upregulated in the MIR21 knockout, but only some of them were common. And the same happened in the downregulated genes. And what are the processes involved in these upregulated and downregulated genes? We observed that some of the pathways were more activated in wild-type mice, such as the pathways in cancer, PA3 kinase activity, the VEGF signaling pathway, the RAS signaling pathways. So a lot of the pathways involved in the development of HCC were more increased in wild-type mice and reduced in the MIR21 knockout mice. And the same when we evaluated gene ontology processes, where we evaluated the cell migration, cell cycle, that these processes were reduced in MIR21 knockout. So it seems that the MIR21 knockout mice display a less oncogenic environment that results in the prevention of tumor development. We confirmed that the proportion of Ki67 positive cells were increased in wild-type mice fed with the CDA, while the levels of these nuclei were reduced in knockout mice fed with the CDA diet. So the hepatocytes in the knockout mice were less proliferative. And we confirmed this by measuring the levels of several oncogenes and tumor suppressors, such as P21, BOX, and the levels of active caspase-2 that were modulated in the wild-type mice, particularly in nodule-enriched liver pieces, while the levels of these proteins were restored to basal levels in the knockout mice. The same happened with the AFP, which is the well-known marker of HCC development, which was substantially increased in the wild-type mice and reduced in the knockout mice. Finally, we evaluated the levels of this pathway in human HCC, And to do that, we use the TCGA cohort, which is comprised of 50 surrounding livers and 362 samples from patients with HCC. And we were happy to observe that ME21 expression was markedly increased in tumor samples from patients with HCC when compared with surrounding liver, resulting in a marked decrease in PPAR alpha levels. And importantly, the levels of PPAR alpha negatively correlated with ME21, which indicates that PPAR alpha might be directly targeting by ME21 in HCC patients. We also observed if we stratify our cohort in high and low expression of both ME21 and PPAR alpha, there was a clear correlation with overall survival, with patients with high ME21 levels displaying worse overall survival, and on the other around, patients with PPAR alpha low levels display the worst overall survival. We also correlated the levels of ME21 and PPAR alpha with well-known markers of HCC progression, such as AFP and LIPCAN3, and we observe a positive correlation for ME21 and a negative correlation for PPAR alpha. So as a conclusion from this fourth part, we can say that ME21 is not only increasing patients with NASH, but also in NASH-associated HCC. And we observed that the ME21 knockout mice displayed a marked reduction in inflammation, steatosis, and were almost completely protected against tumor development. And the genetic correlation of ME21 restored some carcinogenic related pathways, therefore impacted in the progression of NAFLD to HCC. So in the end, I would like to thank the Cell Function and Therapeutic Target Group and the Liver Disease Diagnostics and Therapeutics Group in IMED in Lisbon, where this most of this work were done. My two PhD supervisors, Cecilia and Rui, my lab mates, Marta and Andrea, which were mostly involved in conducting this work, and also our collaborators from the University of Lisbon, and also my group in Biodiagnostic Health Institute, our liver diseases group, which is led by Dr. Jesus Banales. And I would like to thank also all the people involved in this work while I'm doing my postdoc here, Dr. Luis Bujanda, Dr. Raúl Jiménez Agüero, and Laura Izquierdo Sánchez. And I'm happy to answer any question, and thank you very much for your attention.
Thank you
very much. Just remember that you can ask your questions in the chat from the YouTube and also in the chat from the Zoom.
Perfect.
Okay?
Yeah.
Just for the audience. Okay, so Patricia, because you are going to.
Yes, thank you very much, Pedro. This has been really a nice story. It begins from the initial stages of the disease and goes to cancer. And well, you have tell us about really important results regarding lipid metabolism, regarding inflammation, regarding fibrosis. So, well, I have lots of questions, of course. So I just want to, well, if we don't have questions by the moment, I have some of them. So I'm going to ask you, well, curiosities, and I think that there are like really important points here. So well, since you show that miR-22 plays this role, as I said, in lipid metabolism, inflammation, fibrosis, cancer. So is this miR-21 expressed, does it express in hepatocytes and in other cell types in liver? What do you know about this?
That was actually, thank you very much for your comments and for your question. That was actually one of our questions. We know that miR-21 is expressed in all of cell types, both in hepatocytes, Kupffer cells, stellate cells, and cholangiocytes. We know that this microRNA is expressed in all cell types. And we derived our hypothesis thinking that miR-21 would have a more prominent role in hepatocytes. At the same time that we were doing our work, there was another paper coming out also evaluating the role of miR-21 in NASH. And they observed, when they compared patients with steatosis and NASH, that the cells that were mostly presented an upregulation of miR-21 were inflammatory cells and cholangiocytes. So in these patients, although hepatocytes express MAR21, the increased levels were particularly observed in inflammatory cells and biliary cells. And actually, we know that if you evaluate MIR21 expression in mice without NASH, if you only get samples, for example, from OB mice, that they have a huge steatosis, but they don't have inflammation, MIR21 is actually reduced. So it seems that MIR21 is increased when an inflammatory state is evident. But then we thought probably the important cells are the inflammatory cells. But then another study came out where they compared the phenotype of a global knockout of MIR21 and hepatocyte specific, and they observed that the phenotype was the same, which indicates that the MIR21 in hepatocytes also have an important role. And they observed that particularly in lipid metabolism and insulin resistance. So we know that it's expressed in all cells. It might be more prominent when there is a clear inflammatory state, but I think that it has a role in all types of cells. Because, for example, in hepatic stellate cells, it is known that MIR21 is involved in the process of activation of a quiescent hepatic cell to a myofibroblast that might promote also fibrosis. And in Kupfers, we know that MIR21 has an NF-kappa-B responsive element, and it might produce an inflammatory response in Kupffer cells. So in sum, I think it has a role in all cells.
Okay, so I think Isabel has a question.
Yeah, congratulations for the talk. It has been really interesting. It has been a while I have not been working in liver, but still I find it is very fascinating and of course very important from the pathological point of view. So maybe because I also was working in insulin and now we're working on IGF-1, and they share a lot of a lot of common downstream substrates and actions, some of them with different tissue specificity, etc. But for example, in the muscle that you have also shown data there, the IGF-1 system is very important. And I'm wondering if it is regulated in some way. You have to check this. Whether there is an activation of the IGF-1 receptor or in the muscle, because in the liver it's supposed not to be expressed, at least in physiological conditions. But in cancer, if I remember it right, in cancer there was an upregulation of the IGF-1 receptor. I don't know if in this other situation.
Thank you very much for your question. Regarding the IGF-1, we have not focused on that pathway. We only focus on the traditional insulin pathway. I would say that the IGF-1 probably would be activated in NASH because it's well known to be to promote the growth of hepatocytes and also more prominent in muscle. What we observed is that in our CDA model, the one that NASH progresses to HCC, in our mouse liver cancer array, we observed that not IGF-1, but one of the hits of the array was IGF-BP3, I think, which is insulin growth factor binding protein 3, which was the most deregulated protein in the MIR21 knockout mice. It was like 80-fold expressed compared with the wild-type mice. And we observed that this protein, the IGF-BP3, is usually downregulated in HCC. It has a role in counteracting the IGF pathway. And we thought probably this huge increase in the MIR21 knockout mice might also be related with the inhibition of the progression to HCC. But it would be interesting to check also the IGF-1 in muscle. We will take that note.
Yeah, okay, no, that's a very interesting observation. It's indeed suggesting that there is a regulation in the liver. I was, as I said, if I am allowed a second question, do these mice, have you ever checked if they have a neurological phenotype?
A neurological? As far as we know, we have been working with all of them. We we have not observed any differences in their behavior. They are leaner, but no differences in their behavior. And in the other works, well, they did not use knockout. They used an inhibition of MIR21 with an antagomer. They also did not reported any changes in their behavior.
Mm-hmm. Okay, thank you, Pedro.
Okay, and to link with the antagomer action, do you think that treating with the antagomer in hepatocellular carcinoma, for example, will also provide advantages in front of chemo treatments, or what do you think about this? Chemotherapy, or?
I would not say advantages. I think I would say they should be used together. I'm not sure if using, because we know that MIR20, the MIR21 is being studied as an oncomer for 2 or 3 decades now, and so far we have no evidences that MIR21 alone is being studied as a therapeutic target for cancer. Probably because alone it's not enough to inhibit cancer progression. I would say that if we combined MIR21 inhibition with chemotherapy or even with other targeted therapies, we'll be able to achieve a better therapeutic response. And not only to achieve a better therapeutic response, but also probably to surpass therapy resistances. Because probably we can chemosynthesize cells.
Exactly. Yes.
For example, for the targeted therapies, there are a lot of patients that display sorafenib resistance that are usually related to the activation of MAP kinases downstream of the targets, and probably since MIR21 targets several MAP kinases pathways and touches several points in this network, the combination of both might have a synergistic effect when they compared with their both therapies alone.
And probably it's better working on prevention to progression of the disease, no? That in the disease in the tumor itself, no?
I think in the future, if we consider the NASH patients, I mean there are, as I mentioned, there's a huge prevalence of NAFLD. Only a small around 20-25% of the NAFLD patients progresses to NASH, but still, since we have a huge prevalence of NAFLD, we have a marked number of NASH patients worldwide. We should, in the future, try to develop screening programs in patients with non-alcoholic steatohepatitis, because probably there will be the NASH is currently now considered that in the future it will be the first indication for liver transplantation. So it's clearly gaining a lot of relevance in the clinical society. It will be difficult to develop screening programs for everyone because there will be a lot of patients with NASH probably, but the prevention is the key now, because if we have a patient with advanced HCC, and right now with the approaches we have, it's difficult to properly treat these patients. So prevention is the key, that's for sure.
And Pedro, I'm curious, which is the physiological role of MIR21? Thinking in using as a pharmacological target, and thinking also in the side effects that we can observe when we use this target.
Basically, it regulates cell cycle and cell proliferation, and it evolves in the first stages of embryogenesis. Regarding the side effects, I would say that for MIR21, for MIRs in general, to treat liver diseases, we are quite lucky because there's a problem that is still not solved, which is the first passage effect in the liver. Most of when we administered anti-nucleotide oligonucleotide senses for MIR21, most of them are retained in the liver. So for us, it's good because we want to treat the liver. That actually the only, or mostly the only MIR-based therapy that is currently being tested in clinical trials is an anti-MIR122 for hepatitis C. I would say that probably indeed in that case, since most of them is retained in the liver, the side effects will be lower. We still have to consider the potential side effects by acting only in the liver on healthy cells, But as far as we know, when we injected, because we were conducting another study where we treated mice with an integument instead of using the genetic ablation, and we did not observe any liver injury or whatsoever in the control mice treated with the integument. So for now, we believe that will not be substantial adverse effects.
Is that good news?
We have a question here from the audience. It says, thank you so much, Pedro, for your seminar. Very interesting. The semi-synthetic polyuracid you used would also function as the microRNA knockout in protecting liver from steatosis and cancer?
So as far as I understand, they are asking if the obeticholic acid also protects from steatosis and cancer. From liver steatosis, it's described that obeticholic acid protects mice and humans from it decreases the accumulation of fat. That's why obeticholic acid is now in phase 3 clinical trials. There was a huge excitement with obeticholic acid because it was thought that it could be the first therapy approved for these patients, because until now we don't have, rather than sports or changes in lifestyles and dietary habits, there are no pharmacological therapies to treat these patients. And obeticholic acid is now regarded as one potential therapy from NASH. One concern regarding obeticholic acid is it has very good results in treating NASH, but the concerns on the long-term safety are being evaluated, because we know that FXR is modulates bile acids, it's anti-inflammatory, but obeticholic acid also activates the FGF19 pathway in the intestine. That FGF19 goes to the liver to promote the FXR related targets. And we also know that FGF19 is also mitogenic. It might promote hepatocyte proliferation. So until now, we have no evidences that it might promote cancer. So we think that by inhibiting disease progression, it also alleviates the progression to HCC. But we don't know what happened in patients that might take obeticholic acid for several years. There are some concerns regarding that.
And what about urso, the oxycholic?
Well, I think ursodeoxycholic acid was a little bit of disappointment in NAFLD. In the beginning, it was thought that because ursodeoxycholic acid is being used for the treatment of primary biliary cholangitis and intrahepatic cholestasis, for example, in pregnancy or gallstones, it has very described roles as cytoprotective and also in promoting cholerasis. But in NAFLD, there was no changes in these patients. There were several clinical trials already conducted. The clinical community was really motivated with using urso for these patients, but most of them did not present any positive results in the clinical trials. And now urso is not being considered for the treatment of NAFLD.
Okay.
Yeah.
Okay. Well, thank you very much. It has been a great talk. I think it's time to leave, right? And well, not just the talk, but also the discussion. It has been amazing, Pedro. Thank you very much.
Thank you very much.
Yeah, and hope to see you soon.
Oh, I hope we can meet all together. And thank you very much again for this kind invitation and for this interesting discussion. It was really a pleasure.
So thank you, Pedro. Thank you, Patricia, also for organizing the webinar, and Isabel for being here with us, and all the audience for attending the webinar. We will see in the next webinar, and thank you to all. Bye.
Thank you. Bye.
Bye.